Hello, Science 30. This lesson is on genetic or DNA technologies. And in this lesson, I'll talk about half a dozen or so different technologies that you will need to know for Science 30. So the first one that I'll chat about here is DNA fingerprinting. DNA fingerprinting, they've been using this technology for, since the early 1980s. There have been multiple different changes. In your textbook, they give you one version of it where they use what are called restriction enzymes to cut the DNA. I'll give you more of an updated version here on DNA fingerprinting, also referred to as DNA profiling. So it is based upon the fact that there are no two individuals in the world, unless you have an identical twin, that have exactly the same sequence of bases on the chromosomes, the DNA, the A's, the T's, the G's, and the C's. So the analogy is, well, no two people have the same actual fingerprint, and no two people for sure have the same DNA fingerprint, although DNA fingerprinting would be much, much more accurate than the old school fingerprinting techniques. What they do take a look at in the DNA is what are referred to as the non-coding regions. So the coding regions of the DNA, those are the sections of the DNA that provide instructions in order to make proteins. And if we take a look at those sections of DNA, the coding DNA, it really doesn't vary that much between you and me or anyone else. We all have pretty much the same instructions, for example, for making that oxygen transporting protein hemoglobin that you do find inside of the red blood cells. So the non-coding regions, in fact, it's only about 5% of your DNA that is actually coding DNA. A huge percentage of it, well, we don't know what it does, and what we do know is that it's highly variable from person to person. And one of the reasons why it is variable is there are sequences of bases that don't seem to code for anything, but they've been copied or repeated multiple different times. So for example, we could have a sequence of bases. I'll just sort of randomly write down some sequences here. We can have a sequence of these bases, and it could be repeated multiple different times on a given chromosome in one person, and that could differ in another person. So these sections of DNA that are repeated multiple different times, they go by a couple of different names. So this one right here would be a VNTR, a variable nucleotide tandem repeat, or they also take a look sometimes at some shorter ones that are only a few bases long. So maybe one like GAC, and this would be what is called a short tandem repeat. In both cases, though, they've just been repeated many, many different times. So remember that every individual in the nucleus has two copies of each chromosome. One came from the mother, one came from the father, and of course they're not identical. So that means they would also have a different number of VNTRs or STRs on the two chromosomes. So the picture that we see at the bottom here it is showing VNTRs, and we have this sequence here of these bases, and they say they range between 17 and 40 bases in length, the repeat length, which might be repeated, well, many, many different times. How many times do we see here? So again, this is a diploid cell because we have two of the same chromosome, or at least the same number. So this might be chromosome number 5 that we're taking a look at. This one may have come from the father, and this one at the bottom here may have come from the mother. But they're still both chromosome number 5. But if we take a look at this chunk of DNA, the VNTR, or again, it would be the same thing with the STR, what we see is that there are a different number of chunks or repeats on the chromosome that came from one parent. In this case, they're saying 12, compared to the other parent, which is 17. So if we were to go to me, take a look at my cells, take a look at my chromosome number 5, maybe on one of mine, I have 7 repeats, and on another one, maybe I have 18 repeats. So even by taking a look at just one of these STRs or VNTRs, we might be able to see that there's a difference between individuals because this is a highly variable region of the DNA. So before we can start and go ahead and actually do the DNA fingerprint, typically if we're talking about something like a crime scene investigation for sure, we have a very, very small sample. So that can come from various different sources. So from TV shows and movies, you might know that they come from things like blood stains, saliva that's on the cigarette, from semen, various different sources where the DNA can come from. So they do want to make a whole bunch of copies of the VNTR, and there just might not be enough in the very, very small sample that they have to start with. So what they do is they're going to make a whole bunch of different copies, of these chromosomes using a technique referred to as the polymerase chain reaction, or PCR. It's essentially DNA replication in a test tube. And over the course of a day, what it allows you to do is take a section of DNA, which you maybe only have one copy of or very few copies. And at the end of the day, they go through 35 cycles as they have here of amplification, which is really just replication of the DNA in the test tube. We end up with 68 billion copies. So now there is more than enough to go ahead and actually do your DNA fingerprint. What they're going to do is they're going to take the samples that they have what we call amplified. They make many, many copies of the VNTRs or STRs, and then they're going to put it on a molecular sieve, which is referred to as a gel. So this is, in fact, kind of a jello-like material, and it's going to separate out molecular things like DNA based upon their size. So the way that it works is you would put sample at the top, the sample of the DNA in different columns, different sample coming from different people perhaps. Then you would turn on an electrical supply. DNA has a negative charge, and it's going to migrate through this jello-like material. So if we have very small DNA, then they're going to travel quite far. If we have large pieces of DNA, they don't travel very far at all. And other ones that are of sort of middle length, well, they'll end up somewhere in the middle. So again, we're separating the DNA based on size, and that is what the DNA fingerprint is really based on. In fact, this is a DNA fingerprint that we are taking a look at here. This is what it does in fact look like. So kind of going back to, and I'll just talk about the VNTRs here. When they do a crime scene investigation, a forensic study, then they actually look at not one, not two, not even three, like in this picture here. They take a look at 13 different VNTRs. And what they say is that there's virtually no chance that any two humans on the planet are going to end up having the same genetic or DNA fingerprint when they're taking a look at 13 different VNTRs. So here we have 3 different individuals from 3 different people. And what we're taking a look at, it might be 3 different homologous chromosomes, or they could be on the same chromosome. They're just different VNTRs that we're taking a look at. So different sequences of bases that have been repeated multiple different times. So here we have at the top VNTR 1, 2, and 3 at the bottom. And if we take a look at individual A, we can see that for this VNTR on this chromosome that came from one of the parents, there are 3 copies of it. On this one, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, it looks like, what we have there. If we take a look at individual B and C, we can see that it's a different number, even taking a look just at one VNTR. But in some cases, it is actually the same. Okay, so that can happen. For this VNTR number 2, each of them have 3 copies of this VNTR, or 3 repeats of the VNTR. So this is why we don't just take a look at one VNTR, we take a look at multiple different ones. And again, for crime scene investigations, it would be 13 different ones. So now if we take a look at over at the right-hand side here it's showing the homologous chromosomes. So this is really just taking the information on the left-hand side. So if we take a look at individual A, so if we take a look at the lengths of the DNA, this one here is the shortest. And when you put it into this molecular sieve, the gel, because it's the smallest, it's going to travel the furthest. So that, in fact, is this one that we see right here down at the bottom for individual A. Which one is the biggest? This one right here is the biggest, and that's the one that ends up at the top. If we do the same thing, let's take a look at individual C. We can see that this one here looks like that is the shortest, and that's this one right here. And the longest one Looks like it's the same as individual A, and that's the one at the top here. So again, we can't just take a look at one of these because these two individuals have the same. But when we take a look at multiple different VTTRs, you can see that the banding pattern, and that's what we're really taking a look at here. It's like a barcode. We can see that for individuals A, B, and C, even just taking a look at 3 different VTTRs, they don't have the same barcode. They don't have the same DNA fingerprint. So very, very powerful for forensics investigations, and that's what it's showing at the left-hand side here. So this is not really what one of these gel or fingerprints looks like. It's just a representation. But this would be our crime scene bloodstain sample, which they determined came from whoever committed the crime. And now we have our different suspects. So it's really just a matter of this, taking a look at the crime scene DNA, doing the VTTR analysis, the fingerprint for that bloodstain, and then comparing it to the different suspects. So whichever one you have all the bands that are matching up, there's really no question about it, unless there was some sort of sabotage or setting up going on here, John would be the guilty party. He's the only other person in the world, unless he has an identical twin, that has that same DNA fingerprint. It's also used for paternity suits. So over at the right-hand side here, we can see a pedigree at the bottom. So again, the square is for dad, the circle is for mom. We can see that they have 4 children, 2 boys and 2 girls. So sometimes the question is, the father. Is the father really the father of these children? So here they're only showing one single VTTR. And we can see that on this homologous pair here, so it looks like this one is from the father. On one of the father's chromosomes, there are 4 repeats. On the other one, there are 6 repeats. So this would be the DNA fingerprint for dad, just taking a look at this very, very simple DNA fingerprint, taking a look at one VTTR. For mom, we can see that there are 5 repeats here, 3 repeats on the other one. So only taking a look at this one VTTR, mom and dad have a different DNA fingerprint. So of the children that they have, each child gets a chromosome from mom and a chromosome from dad. So they either have to get this one with the 4 or the 6 repeats from dad, and the ones with the 5 or the 3 from mom. It has to be one from each parent. So if we take a look at the boy here, we can see that this band right here, it came from dad. We can see that this one came from mom. This girl, we can see that this one came from mom. This one came from dad. The second female, we can see that this one here came from dad. One at the bottom came from mom. And the last son, this one here, came from dad. This one here came from mom. So according to this very simple DNA fingerprint, all 4 of these children would in fact be the children of these 2 parents that we see here. Societal issues, you do need to know about societal issues for Science 30. So we'll talk about what some of these societal issues are, and I'll give you one example of a typical kind of question that you would get on a quiz or an exam. So societal issues usually fit into what I call different perspectives. So it could be economic. Usually whenever we're talking about money or some sort of cost, that would be the economic one. Sometimes societal and ethical, they kind of split up. Sometimes they keep them together. Cultural, political, ecological or environmental, medical, healthcare, religious, legal, technological, even though this one isn't societal, it's technological rather than societal. They usually throw that in as well as a societal issue. So in terms of DNA fingerprinting, what could be some of the issues? DNA databases. So questions like, should everyone have to donate DNA and have their DNA put into a database? So if something does come up, a crime scene investigation, and then officials can just go directly to these databases. So not everyone is entirely comfortable with having their genetic information out there and available for others to see for various different reasons, sometimes just privacy reasons, and they don't feel that others should have access to that kind of information. Possible discrimination, so we hear about maybe things like employers getting hold of information and finding out that maybe they're susceptible to some sort of different medical condition, or perhaps insurance companies, and they may not want to insure people for healthcare because they have a certain medical condition. We also hear about things like racial profiling as well. So based upon their DNA, they might be able to determine from which ethnic group someone might belong to, and use that information when they're looking for solving a crime. So lots of different societal issues related to any technology, and that is definitely the case with these DNA technologies. So I'm just going to give you an example here, a typical sort of layout of what a question, a societal related question would look like related to a technology. This one actually isn't a Science 30 example. It's talking about hormones, and this one you would not need to know for Science 30. It's just an example though, and even though we haven't talked about hormones at all and don't talk about it in this course, you should still actually be able to answer this question. So a contraceptive implant has been developed for male dogs. The implant releases a drug which is called deslorelin. Now this drug, we have some statements that are related to its use. So administering the drug for a short period of time costs less than neutering a male dog. Remember I said money, costs, quite often economic. So what we have at the bottom here is we have a statement. That's the 4 different statements up above. And what sort of perspective does it fit into? So as soon as we see this cost, you should automatically be thinking, we'll just put this down tentatively. We might have to change it, but already we should be thinking that's economic. Our researchers hypothesize that the drug could be used to control populations of wild animals. So here we're talking about in the environment. So it sounds like that would fit into ecological. Veterinarians are often concerned that the manipulation of hormones with deslorelin will increase the incidence of cancer in dogs. So kind of a risk associated with this technology. So that seems to be a technological issue. And using this drug to decrease reproduction in dogs could decrease the need for organizations like the SPCA and other humane organizations. And that looks like it would fit into societal. Okay, so you may need to sort of debate as to which ones they do fit best into, but in this case here, it seems relatively straightforward in terms of which perspective would go along with each one of the statements. Just quickly on these ones here, the Human Genome Project that we're working on throughout the 1990s and the initial one for the Human Genome Project, the sequence kind of collectively for our entire population, more or less the genetic makeup for us collectively, it was initially completed in 2003. And now we have this bank that we can use to compare our DNA sequence to the other sort of standard and maybe use that for, well, various different things, such as determining if people are susceptible to certain diseases or conditions. So what is the DNA sequencing? Well, it is really just determining what is the sequence, and it's usually not the entire 3.2 billion steps on the 46 chromosomes. It's usually just sections of it that we are sequencing. So typically it's the coding information that is for a gene or maybe some other section of DNA that they might be interested in. So yeah, the key here is that probably medical, useful to determining if someone does have a predisposition to certain diseases and can sort of prepare for that disease sometime in the future, or maybe take some sort of interventions to ensure that they don't end up with that disease, or at least not quite as early in their life. What kind of goes along with that is what are referred to as DNA microarrays, gene chips, or DNA readers. So if we just jump down to the picture at the bottom here, what they're doing is taking samples of someone's DNA, cutting it up into different pieces, adding what's called a radioactive tag, and then washing it over top of a glass plate that has a whole bunch of sequences of DNA. So the A's, the T's, the G's, and the C's, that would be for a gene or usually some sort of a gene defect. So now what they're trying to see is if this person in their DNA, it actually attaches to one of these sections of DNA that's complementary to it, that might give an indication that yes, they do have a defective version of the gene. So that's one way that this could be used for. So this picture here is just kind of showing these different colors, the yellows, the reds, and the greens, and which ones it attaches to, which ones it doesn't attach to. And again, that's typically used or could be used for medical reasons, for healthcare reasons, and determining someone's susceptibility to particular diseases or conditions. This is a big one for Science 30. In your textbook, I believe they use this terminology here. Goes by a bunch of different names, though. I kind of like the first one, prefer the first one, recombinant DNA technology, or rDNA. Or the R for recombinant means you're recombining or joining together the DNA that's coming from two different individuals. So those two different individuals could be two different humans, members of the same population, the same species. They could also be from different species. They can even be from completely different kingdoms. And the reason why this is possible is because every single living organism, whether it's bacteria, fungi, plants, animals, protists, we all use the same genetic code, which is DNA. So it also goes by the name genetic engineering, because quite often we do this intentionally in order to create a useful product. And if we do use this technology to create a useful product, that is genetic engineering, and we'll take a look at a nice example of that. And when you're joining together the DNA, it's just called gene splicing. Splicing is the joining together. And again, as long as it is DNA, you can join it together regardless of where that DNA is coming from. So the first one I have here is genetically modified organisms, or GMOs. So usually we hear about this GMO labeling in terms of foods that we do eat. So plants, they're the big one. There are many plants that have been genetically modified, and they have been genetically modified for certain things like pest resistance, so they can withstand pests in the environment, herbicide resistance, so a farmer could spray a herbicide on the crop and that crop would be able to resist it. So many crops have been genetically modified. Canola is a big one. Corn is another big one that have been genetically modified. Pretty much all of the canola and the corn grown in North America has been genetically modified. But really it can be any trait that we find in any other living organism. And we could say, well, that sounds like a useful trait. We would like to put it into a tomato plant that would maybe allow it to withstand colder temperatures, or allow it to have larger fruit, and that sort of thing. So adding a gene is what we're doing, adding a gene to the genome from a different individual, or maybe even a completely different species. In animals, a few of them that they have tried this with, there's just that one that I'm aware of that's on the market, and that is for Atlantic salmon. And it really just is a gene that's inserted that doesn't allow the salmon to get any bigger, but it allows it to grow at a faster rate. So those would be genetically modified foods in particular that we're talking about. We've done this since, probably going back to the 1970s, genetically modifying bacteria and now also genetically modifying yeast. Of course, bacteria, very different than our cells. They don't have a nucleus. The DNA inside of a bacterial cell, it's not long linear chromosomes like we have, but in fact it's in the form of a circle. So they have a large circular chromosome, and then they also have a bunch of smaller ones, and these are the ones that we're interested in for genetically modifying them. And these smaller ones, they're called the plasmids. Okay, so all of this DNA is inside of a bacterial cell, and that in fact is the picture that they're trying to show at the bottom here. The picture at the top, this could be one of our cells. So we could take one of our cells, and again, this could be from anything, but we do this with our cells all of the time in order to make a useful product and use the bacteria to make this useful product for us. So in medicine, this is used all the time. So DNA containing a gene removed from a useful cell. So what we have done this for humans is the hormone insulin. So people that are diabetic, type 1 diabetic, they can't make this hormone insulin. They require exogenous injections of insulin to keep them alive. And it's actually bacteria that now make that for us. Other things in medicine as well, if someone has hemophilia, clotting factors are also made this way. If someone can't make a human growth hormone is made by bacteria as well. So this is the way it works. So somewhere amongst our 46 chromosomes are all of these genes that code for different proteins like these ones here. So yes, they need to find it, they need to cut it out, and then what they're going to do is go to a bacterial cell, they're going to retrieve the plasmid, and they're going to make a cut in the circular plasmid as well. And then they're going to insert this human gene in this case, which might be for insulin, into this bacterial plasmid. And this is now what we call a recombinant plasmid. So this recombinant plasmid, we now put it back into bacterial cells. Bacterial cells reproduce really, really quickly, maybe once every 20 minutes. So at the end of the day, or definitely the end of the week, you could have billions of bacterial cells that all have a gene that makes a protein that they have absolutely no use for. So now all we need to do after growing those bacteria is retrieve that insulin, bottle it up, and now we have that insulin that we can now supply to people that have diabetes. That is the source of the insulin for people that are diabetic. That is the source for clotting factors for people that have hemophilia, human growth hormone for people that have a deficiency of that, and multiple other proteins that can be produced, human proteins using bacteria. And yeast, they can use yeast for this as well. This is another big one, gene therapy. So here we are specifically talking about humans and genetically modifying humans. So far, really what we're focusing on are what are called single gene genetic diseases. So I'll just give you a couple here, something called sickle cell anemia. Oops, sickle cell anemia. This is what causes the red blood cells to go from the biconcave shape to what's referred to as a sickle shape. They don't transport oxygen quite as well. They get stuck in the blood vessels. Very painful condition. It's caused by a single gene. So if we could get in the correct version, then this wouldn't happen. Another one is cystic fibrosis, very common. This is a single gene disorder. Find the correct gene, insert it into those people's cells, and they no longer have cystic fibrosis. So it involves, yes, replacing the defective version of the gene. You don't even have to yank the defective one out most of the time. You just need to get the correct version in and get it to be read, or what we call expressed. So how do they actually get it in? Well, in this case, what they use is, just in general terms, it's referred to as a vector, but usually it's a virus, because this is what viruses normally do, is they normally infect cells and inject their DNA. So what they do is they go to a virus, kind of like what we just saw with the bacteria, and they put the gene into the virus, they let the gene infect the cell, and hopefully introduce that gene, which is useful, into that cell, and it can now be read. So nice picture here that shows a couple of different techniques. This one is called the in vivo technique, where here we have the therapeutic gene that they have inserted into the DNA of this virus. So this is what's called a retrovirus. And then they inject the virus into the person's body. Viruses are very, very specific for the kind of cells that they infect. So they will infect the target cells. In this case here, they're saying that it's the liver. And that therapeutic gene would only go into the liver cells. And ideally, those liver cells are going to read that information, and then they're going to make that protein that previously was not made by the liver cells. There can be some issues with this. As soon as you inject that virus into the person's body, their immune system will typically attack and destroy it. So for this reason, it quite often doesn't work. Sometimes there will be a significant immune response that can, in fact, put this person's life at risk by doing this sort of thing. And sometimes that DNA that's inserted into the cells isn't inserted into the right place, and it doesn't end up making a protein. Another technique is to actually take some cells out of a person's body, what are called stem cells. These are a reservoir of cells to replace cells in someone's body. So skin cells, for example, you would have skin stem cells that are a reservoir for replacing your cells when they do die, which they do all the time. So take stem cells out, put them in a petri dish. So now they're going to take that virus and allow it to infect the cells in the petri dish. Make sure everything is going okay before you take those cells and put them back into the patient. Okay, so two different techniques in order to do this. So all kinds of problems that have resulted, as I mentioned. So one of them is that quite often the viruses are just attacked. Sometimes they're inserted into a section of the DNA and they interrupt the normal functioning of other sections of DNA. And those two different techniques that we just saw, they are called the in vivo and the ex vivo. This one is in the body, and this is one where it's taken out of the body before those cells are genetically modified. So pretty much all of this so far has been on what is referred to as somatic gene cell therapy. These are your body cells, and that means all the cells except for your gametes, not your sperm, not your eggs. The idea behind this, the thinking is that at most we're just going to screw up the genetics of one person, but at least it's not going to be passed on to the next generation. So this has been tried for multiple different things. There has been some success. There's been some success going all the way back to 1990s. This has a big long name. It stands for severe combined immunodeficiency syndrome, children that are born without an immune system. There was some success with this, and I say some because, as it turns out, some of the children ended up developing leukemia after this treatment. Another one more recently is for, I believe it was hemophilia B. There are different kinds of hemophilia, and I believe they did have some success in treating hemophilia B using gene therapy. Right now, germline gene therapy is kind of a no-no, so it is passed on to the offspring, and because we don't exactly know of the consequences in the future, that's why it's kind of off limits at this point. Again, huge societal issues related to this as well. The ones that I'm talking about, they are therapeutic, where we're actually providing a therapy for someone that does have a medical condition. The other type would be what is called enhancement gene therapy. So here, it's not really needed. It's just maybe you kind of want some extra trait, but it's not for any therapeutic purpose. Last one here is cloning. So cloning of mammals goes back to the 1990s with Dolly the sheep. Cloning of amphibians, in fact, goes back decades before that. Different ways to generate clones. A very, very simple one is just called artificial twinning. So if we do take the zygote, this is the fertilized egg, the zygote. So here, I'll just put in the number of chromosomes in the nucleus for a human. So in theory, we could do this. When a cell divides into two, each of those cells are genetically identical. And then early on, embryonic development is just this division into 4 and into 8 cells. What sometimes happens naturally is these become separated. So if these become separated at this point, then what we end up with are 2 individuals, which doesn't necessarily have to be a female. I'm just going to draw. That's what I want to draw. 2 individuals that would be genetically identical. So this is how you do get identical twins. This is the process. 1 single zygote, 1 egg fertilized by 1 sperm. If we do have in vitro fertilization, which is fertilization in a petri dish, the sperm fertilizing the egg in a petri dish, the first few cell divisions take place in the petri dish before it is implanted back into the female's uterus. So what we could do is we could actually just tease these cells apart up until the 8-cell stage, and each of these cells can now give rise to an individual which would then be genetically identical to all of the others. So there would be no sense to doing this sort of thing with humans, but in fact they do this with livestock. So the idea is you get a prize-winning bull to donate the sperm, a prize-winning cow to donate the egg, and this just bumps up the chance that you're going to get an individual cow or bull being born that's also kind of prize-winning. But wouldn't it be great to just take, or so the thinking is, to have a whole bunch of those and just plant them into several different cows? So cow number 1 gets this one, cow number 2 gets this one, cow number 3 gets this one, and so on and so on. So that is artificial twinning. That does generate clones, but usually when we talk about cloning and cloning the mammals, like they did with Dolly the sheep here in the 1990s, it is referred to as somatic cell nuclear transfer. We're taking a somatic cell, which is a body cell, we're taking the nucleus and we're going to transfer it. And where we're going to transfer it to is to an egg that doesn't have a nucleus. So this picture here very nicely shows it. So this is the sheep here, where we're going to take a somatic cell and we're going to retrieve the nucleus. So this nucleus right here, we're just going to suck it out, or we're going to fuse it with another cell, which is the egg. So this one here is the egg donor. Retrieve the egg, but now that nucleus in here, we're going to remove it. So this is what is called an enucleated egg right here. It has no nucleus. This one here, again, if it were human, it would have 23 chromosomes. This one would have 46 because it's a somatic cell. This one now has none. We fuse it with another cell that has 46. Now we do have the equivalent of a fertilized egg, only we don't have half of the chromosomes coming from each parent. Every single one of these chromosomes came from this sheep here. Again, the number is not 46 chromosomes when we're talking about sheep. I'm just using that number. So now this is what happens in the petri dish. Cell divides several times. We get the embryo implanted, and here they've implanted it into a third different sheep. So here we have sheep number 1. Here we have sheep number 2. Here we have sheep number 3. This sheep here is really just providing the uterus for the growth of the embryo. So this lamb that is born here, what it is going to be identical to is this one that donated the nucleus. Those are the ones that are identical. So they are clones of each other. And again, that goes way back to 1996. Just one last one here, what is referred to as induced pluripotent stem cells. So this only dates back about the last decade or so. So stem cells that we did briefly come across before, they are cells that are in your body, but they are what are called undifferentiated, which kind of means that they don't have a specific function. They're a reservoir of cells, but if we can give them the right signal, what we think is we can turn them into any kind of cell type that we like. So if you do any sort of a tissue transplant, though, then that tissue is typically going to be rejected. So if we can retrieve cells from someone, their own stem cells, and somehow give those cells the correct message to turn them into a specific kind of cell that that individual needs, put them back into the person, then it wouldn't be rejected. But it's kind of difficult to take stem cells that are from the bone marrow and get them to form anything other than the blood cells. So can we kind of reverse the process and turn them into the equivalent of what we have right here, the zygote or the early embryo, which can become any one of the hundreds of different cell types. So that is what they did come up with, and those are called induced pluripotent stem cells. So what they did is they just took an ordinary somatic cell, which could be any cell from the person's body, and they just inserted 4 different genes, and somehow that turned them into the equivalent of what are called embryonic stem cells. And then if they give them the correct signal, then they can turn it into whatever cell they like, and they can use it for a tissue or organ transplant with no rejection. So this sort of overcomes the issue of using embryonic stem cells, because if we do use embryonic stem cells, it does end up, in fact, destroying the embryo. And again, societal issues that would be related to these, related to, is it reproductive cloning that we are doing, or is it for therapeutic purposes? So this would be, of course, for therapeutic purposes, whereas something like this would be reproductive cloning, where we're actually producing another identical individual.